Hi everyone, welcome back to the channel. This is Dr. Hassi and today's topic is Module 1 in Bio 2 1011 Lab. And this is just a review of just the 5 chapters that we actually cover in Module 1. And let's get started. So we start with phylogeny, which is part of systematics. So in the lecture, you learn about systematics. The whole class uses systematics, which is combination of taxonomy, which is classification of organisms, and phylogeny, which is the evolutionary relationship between the organisms, right? So some of this stuff we know from their fossil records, remaining traces of the past organisms. Also, we see some environmental changes and how populations evolved and change in these the various organisms. So one thing that we use a lot in this kind of evolution is homology, which is basically homologous structures that pass down from generation to generation. It's basically due to similarity due to ancestry, okay? Like genetic code, all organisms uses DNA, right? But we also see genetic variation, which makes the evolution possible. And you see here this clock on the left here, which 4.5 billion years old clock. And there are different zones there and eras, and starting with the right side, like archaeans, and we see 3.5 billion years prokaryotes appearing, 3.4 billion first photosynthetic for prokaryotes, and then we see oxygen starting 2.2 billion 2 billion years, and then we start seeing actually very first algae maybe 541 million years, and then first plants 444 million years ago, then animals, and then more plants, gymnosperms 200 million years, angiosperms 145 million years ago, and then human maybe 2 million years ago, and then to the current present time. So it ends in the present time in that clock. So on the right side, we see a phylogenetic trees. Phylogeny shows evolutionary relationships. There are branches, is like any tree. There is a root, which basically most recent common ancestor, and then all these branching points, and each branch we can call taxon. It could be taxon A, taxon B, taxon C, taxon D, taxon E, and goes on like that. There's a basal taxon which goes from the root to all the way to the end without branching out. And there are sister taxons, maybe they like branching out from the very end, that they are closest to each other, and then branch points that we have. And this is again because of this whole evolutionary relationship, we can actually design this tree called phylogenetic tree. So just remember that. So this next lab was about starting really the kingdom Protista. These are called protists. These are organisms that they are unicellular eukaryotes, protists, right? So they might have plant, animal, or fungi looking organisms in this. So anything that eukaryotes minus plant, animal, fungi, they are actually placed in this, listed under this this group called protists. So protists have 5 supergroup. In this picture, you can see here different groups like slime molds, amoeba, euglena, diatoms, macroalgae, dinoflagellates. These are some representative species under this kingdom Protista. This is lab 3. Lab 3 and lab 4 basically both cover Protista. So 5 supergroups. Supergroup excavata, supergroup chromalvallata, supergroup rhizaria, supergroup archaeoplastida, and then supergroup uniconta. Okay, so 5 supergroups. So I have always start with PowerPoints. PowerPoint slides. They are in your canvas under module 1 in this case. So at the end of each PowerPoint, there is a table there that you worked on when basically when you were making your drawings in your notebook or iPad app. So look at those, and this is like a list of lab 3 drawings. You can see all these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different drawings. These are, you need to be familiar with these drawings and what their names are and what's happening. So besides, also you should look at your lab mini lecture notes that I cover at the beginning before you start drawings. So some little story about each of these species. Okay, so what you must recognize them. That euglena, this is the euglena, this is standard, this is diatom. And so these are from your drawings, either mostly from microscope slides, but also some of them are from specimens or dissecting pen or specimen is jar that you actually looked at. Okay, so make sure you look at that. Excavata is our supergroup 1 under Protista. They have subgroups Diplomonadas, Parabacillates, Euglena zones. Euglena, obviously, Euglena is one species, very important here, which is you need to pay attention. The flagella number, which is single flagella, that at least known, the most well-known one. The next ones are Chromovalada, like we see diatoms in that supergroup, and also brown algae, and also some Oomycetes, Dinoflagellatus. These are all under supergroup 2. Supergroup 3 is Rhizaria, which is Amoebas, for example, a species. Supergroup 4, Archaeoplastida. Red algae, green algae, and land plants are under the supergroup 4, Archaeoplastida. And supergroup 5, Uniconta, which is slime molds, fungi, animals, they are listed under this. We will keep seeing this more in the future, so just study this. And so the rest of the supergroups are here in lab 4, which basically red algae, and you can see here brown algae and the green algae. So all these groups are here. So make sure to go over that. Okay. Now, and then lastly, we seen we start bryophytes in lab number 5, which is actually Plantae. Plantae is a kingdom itself, which is whole plant, more than 300,000 species actually listed on the Plantae kingdom, a very cool kingdom, which has several phylums, like or groups first, like non-vascular plants that we see in here, bryophytes. Then later in the labs that we will see seedless vascular, we will see gymno and angiosperms. So the lab 5 covers only bryophytes, which is, and also cover only 2 phylums, mosses, which is Bryophyta, and then the second phylum is Hepatophyta, which is liverworts. Mosses have stomata, 12,000 species, and a good example species is Minium, M-N-I-U-M. Okay, so again, both of these guys have Antheridium, which is male, Archegonium, which is female, with the egg. Again, Minium is a moss, but there are other mosses like you can see in this picture here, Sphagnum and Polytricium, which is some other examples. Liverwort is a phylum, Hepatophyta. Hepatophyta, a good best example is Marchantia. Marchantia, you can see here, Marchantia is a liverwort species. There are other Marchantia, other liverworts like Richia, Pelia, and some other examples. Liverworts don't have no stomata. They also have an asexual reproduction called gemma cups, like this guy here, which is a gemma cup. So check your notes on gemma cups. They also have Archegonium, female with egg, Antheridium, male with sperms. So make sure you check those. Gemma cups is non. It's not the fertilization base, but more asexual base. Okay, so reproduction here is done by spores. So, and this is, they came up 470 million years ago. So this is sporophyte is 2N and gametophyte is 1N. So make sure check their life cycles. So this is lab number 5. And with all labs from 1 to 5, check the tables at the end of the lab. So items that you're supposed to draw, look at your notes and your lecture notes, lab lecture notes, and then look at your drawings and check every drawing and make sure that maybe you make flashcards of these drawings and identify which is which in 1 to 5. So exam is going to have a lot of images and asking you things related to each other and all individual ones. Gametophyte is always N1N, and we are going to see more gametophytes in the future. In the early plants, they are very dominant, unlike later, most recent plants. So, just to recap, this was module 1, and we are going to have a more review in class 2 and a quiz, and then this is a review for exam 1 in BSC1011 lab module 1 coming up. So please feel free to comment and subscribe to the channel Dr. Rasi Explains, and see you in next video. Happy studying.